Rahim. Today I'm going to talk about cell cycle and how replication of cell is regulated. So let's start. By end of today's lecture, you should be able to understand cell cycle and its regulatory mechanism, its different phases, how cell proceeds through these phases, and what are the checkpoints and their importance in regulation of the cell cycle. Proliferation of a cell is a tightly regulated process in which many molecules and interrelated pathways play their part. Remember, whenever a cell has to divide, first it has to be stimulated. And this stimulation is done by certain growth factors and signals from the extracellular environment. This whole series of events is tightly controlled process, and this is known as the cell cycle. The cell cycle has three stages, which are quiescent, interphase, and mitosis, and five phases: G0, G1, S, G2, and M phases. The quiescent state is also known as senescent state, and it is comprised of G0 phase. The interphase is comprised of G1, S, and G2 phase, which is shown here in green color. The mitosis state consists of M phase, and this M phase is further subdivided into prophase, metaphase, anaphase, and telophase. This M phase here, it is shown in gray color. You all already know about these mitotic phases, so I think there is no need to go in detail. So when a cell passes through cell cycle and these phases, first it has to present itself to certain checkpoints. You can relate these checkpoints to screening devices. So what these screening devices will do? These screening devices will identify any kind of damage or abnormality in these dividing cells. Once these dividing cells are cleared of any abnormality, only then they will be considered as totally fit and healthy, and thus they can progress through other phases of the cell cycle. It is important to note that the checkpoints and these phases are controlled by many genes, and in addition to these genes, there are certain activators and inhibitors responsible for progression of cell cycle. When the cells are in quiescent phase, they have to pass through the G0 to G1 through activation of the specific genes. This acts like a gateway to the entry of the cell cycle. Restriction point is denoted by R, and it is a point in G1 phase at which the cell becomes committed to cell cycle, and after which extracellular proliferatory signals are no longer required. Checkpoints. There are three major checkpoints which are denoted by G1S, G2M, and M phase. At G1 checkpoint, it will be checked whether the cellular DNA is okay or not. Are there enough resources and proteins for replication of the cells? And is the environment suitable for replication of the DNA? At G2 checkpoint, it will be ensured that DNA is correctly replicated. It will also check whether the DNA replication is completed or not. And also, it will be checked that are there enough proteins in the cells to enter into the next phase of the cell cycle. M checkpoint. At this checkpoint, cell will be scanned for proper spindle formation, correct orientation of the chromosomes, and it will also be checked whether all the chromosomes are attached to the spindle fibers or not. So this figure gives a brief overview of where does these checkpoints operate and what are their main function. At G1S transition, there is a checkpoint which is shown by a policeman with a stop sign, and it will check the integrity of a DNA. At G2M transition point, there will be scanning of the DNA replication. And finally, at the metaphase to anaphase transition, the chromosomes will be scrutinized for proper alignment. Progression through checkpoints is regulated by some proteins, cyclins, and then enzymes, cyclin-dependent kinases. During G1 phase, Cell cycle progression is controlled by some different cellular mechanisms such as the tumor suppressors, genes, proteins, enzymes, and signaling molecules. While passing through G1S checkpoint, certain proteins known as the cyclins and as I have already told you, and their related enzymes CDKs, they will start regulating it. So what these cyclins and CDKs will do, these CDKs will bind cyclins and thus they will form a complex. And in this way, they will be activated. As shown here in this picture, the red ones are the CDKs, cyclin dependent kinases, and the blue ones are the cyclins. Upon their binding, a complex will be formed and thus they will be activated. So now what this complex will do? This complex will phosphorylate proteins which are essential for cell cycle transition. I will try to explain this process with the help of an example. Many of you might have heard about the RB protein. This is a tumor suppressor protein. This means that it will not allow the cell to proliferate unnoticed and will keep their eye on dividing cells. So the RB protein, it will make a tight inactive complex with a transcription factor known as E2F. So now a specific cyclin and the CDK complex will phosphorylate this RB plus E2F inactive complex. After phosphorylation of RB, it will get released from E2F, the transcription factor. And in this way, the E2F transcription factor will be activated. Upon the separation of E2F from RB protein, the E2F transcription factor will go into the nucleus, and it will start affect the genes. Certain genes will be they will start expressing themselves, and as a result, certain specific proteins are made. And these proteins will thus push the cell into the next phase of the cell cycle. In case of DNA damage, so what happens when the cell's genetic material is damaged? In that case, the repair machinery of the cell will come forward and it will arrest the cell cycle. And at the same time, it will try to repair the DNA. However, if this damaged DNA is beyond repair, then the apoptotic mechanism will be activated and the cell will be finally destroyed. So students, this is the end of today's lecture. Cell cycle regulation is a very important topic, not only from the examination perspective, but also in order to understand how the cell escape this regulatory mechanism and thus they can lead to formation of malignancy. I hope I have made it easy for you to understand. Thank you so much. Allah Hafiz.